Well, first things first, what are enzymes? Well, enzymes are catalysts that reduce the activation energy of a chemical reaction. Most enzymes are proteins. We all have enzymes in our bodies. As we have been discussing, we are trying to isolate enzymes that can degrade cellulose. So in our project, we used a lot of media. For example, we use liquid media, LB liquid media, which is rich in nutrients. And we did this because we needed a lot of cells, because we needed a lot of protein from those cells that can degrade cellulose. So we managed to grow a lot of cells, but the thing is, the bacteria got lazy, as in it didn't need to produce cellulases anymore because it had the rich nutrients. So it automatically shut down. So we plated them on powder plates, the cells on powder plates, cellulose powder plates, so it can produce those enzymes again. Okay, this has something to do with my project. Tell you. Okay. In the bacteria, some bacteria has enzymes that are extracellular, which is basically an enzyme that's outside of the cell. It's not inside, it's out. So cellulose is a very rigid structure, as Martine stated earlier. So what the bacteria had to do is take these little friends over here, enzymes, and break them down. So it's just like this. We can't eat a whole cow even if we wanted to. So we got to break it down, you know how that goes, till finally we can eat it. The same thing for the bacteria. Which makes it good for us because we wanted to filter, we wanted to isolate the enzyme. We did this by filtering. So it's basically like, I'll show you. It's basically like we scraped the powder that had the cells growing the cellulase, and then we placed it in a filter, just trying to get the enzyme and leaving the powder in the bacteria. So it's like for those who like orange juice without pulp, we filtered the orange juice and left the pulp. The powder and the bacteria is the pulp, and the orange juice is the enzyme. So just like in school, we get tests, right? They test us to see if we're good in math or in any subject. Well, right here we're trying to test the enzyme now, and that's an enzyme assay. Basically, is to test the enzyme. So an enzyme assay is a laboratory method for measuring enzyme activity. Our cellulose was dyed and it was attached, every 20 monomers were dyed with this dye right here. There we go. And then we just wanted to see if the enzyme can cut cellulose, as shown. We found out that the enzyme could cut the cellulose since the cellulose is insoluble. You notice here, the blue is the cellulose. It's not really like this one. I'll show you right now. Since the cellulose is insoluble, we know that it worked if the enzyme cuts the cellulose down, because the monomers will float up, and you can see that it becomes soluble by color death. The darker it is, the better for us. We noticed enzyme activity with our protein that we were working with from our bacterial strains, and we didn't even purify it, so that's good. We also ran the gel. A page gel is a technique to separate and characterize proteins. We used this gel to evaluate our proteins, and we found out that our proteins are about 33 kilodaltons. We will use chromatography now to purify our gel, or to purify our protein, sorry. So once our protein is purified and ready to go, we're happy. And we will we will send it for sequencing so we can get further down in our project. And I will pass that to my dear friend Vernon.
Okay. Hello, my name's Vernon. Good afternoon, ladies and gentlemen. I'll be talking about how to turn a protein into a gene and why we are going to do that. So first of all, we need to know what a gene is and what a protein is. A gene, for instance, is a strand of DNA, as you can see in this picture to the left. It's a strand of DNA that holds information, like a recipe, to make proteins. And DNA in complex organisms, such as humans ourselves, is more complex. We have these exons and introns, as you can see on the left. E. coli's gene map is on the right. You can see that it's sort of, well, no you can't, but I'll just tell you this. It's less complex than our human genome because humans have about 20 to 25,000 genes and about almost 3 billion nucleotides, whereas bacteria such as E. coli have a few hundred genes and a couple thousand base pairs worth of DNA. This image shows the process of transcription and translation. This is how DNA is read and turned into a protein. The enzyme RNA polymerase reads a part of the DNA. In this case, it's a prokaryote. So the enzyme will read the portion of the DNA that codes for this protein you can see here in blue. The enzyme will make an mRNA strand that you can see in red, which is sort of the recipe booklet that you can use when you cook or something. A ribosome will just take the recipe and follow the instructions and make the protein, which here is shown in blue. I'm going to define a protein now. So after those two processes are finished, we will get something that looks like this. Proteins have many different shapes and sizes. They're all used for different functions. Proteins can either be used as structural bricks, or they can be used as enzymes that do a specific job. So after the ribosome creates the strand of amino acids, which folds into this protein. It just folds into this protein. Not much to say about this. So mass spec is how we will begin to turn the protein into the gene. Proteins are fairly large organic molecules, to say the least, compared to the substrate counterparts. So we introduce proteases to chop up the protein into smaller pieces, ideally around 7 amino acids long. Then we shoot it through this metal capillary, and depending where each molecule lands, the computer, using a series of complex algorithms, will list for us, hopefully in the correct order, what is the amino acid sequence for the chain, for the small fragment. After we get the amino acid fragment, we will turn the 7 amino acid fragments into primers, which we will use for PCR. As you can see in the